Welcome to the premiere of Across the Rheum, the official podcast of Rheum Connect, where cutting-edge science meets expert clinical insight in the field of rheumatology. I'm your host, Dr. Sergio Schwartzman, and I'm honored to launch this new educational series with a three-part titled The Interleukin-23 Paradigm in Psoriatic Arthritis. In this series, we'll take a structured look at how interleukin-23 has emerged as a critical target in the treatment of psoriatic disease. Our discussion will unfold across three focused episodes, including the immunology of interleukin-23, a review of currently available interleukin-23 agents, and the clinical utility and positioning of IL-23 inhibitors in today's treatment landscape. Today's segment marks the beginning of this dialogue, featuring our distinguished guests, Jessica Farrell, PharmD, and Dr. Monica Schwartzman. I am Dr. Sergio Schwartzman. I'm a rheumatologist in New York City. I've been in rheumatology for approximately 4 decades now and have split my time. My first 3.5 decades were in academia at the Hospital for Special Surgery and at Cornell Weill Medical School and the New York Hospital. And over the last 6 years, I've been in private practice in the Upper East Side of Manhattan. My research interests, which are a reflection of my clinical practice currently, have been in autoimmune eye diseases, in psoriatic disease, the axial spondyloarthritis group of conditions, because it is an umbrella, and in inflammatory bowel disease-associated arthritis, which is a new interest. Jessica Farrell, could you please give us a little bit of an introduction into who you are and what you do?
So thank you for having me. Yes, my name is Jessica Farrell. I'm a clinical professor in the Department of Pharmacy Practice at Albany College of Pharmacy and Health Sciences, and also a clinical pharmacist at Albany Medical Center, Division of Rheumatology, in both the adults and pediatric clinics. I've been in rheumatology for 17 years now. And as part of the team, I work in clinic with the rheumatologist. I help them make treatment decisions based on patients' disease characteristics, comorbidities, access to medication. I do a lot of patient education and counseling, and I'm really happy to be here today. Thank you.
Dr. Monica Schwartzman, can you provide a brief introduction?
I'm excited to be here. Thank you for having me. My name is Dr. Monica Schwartzman. I'm a rheumatologist in practice. I've been in practice a little less than the other two speakers here, about 4 years. And I did my medical school at Mount Sinai School of Medicine in New York City. And I did my internal medicine residency there as well, followed by a rheumatology fellowship at the Hospital for Special Surgery. And over the last 4 years, I've been in private practice with my dad in New York City. And I'm also a clinical instructor at Weill Cornell Medicine, New York Presbyterian Hospital. I have a clinical and research interest in the spondyloarthritis group of diseases, and I'm excited to speak with you all today about this interesting topic.
Thank you both for the introduction. I also wanted to note that this episode is being cross-posted on the Arthros website and the Rapcast, the official podcast of rheumatology advanced practice providers. And we would like to extend our sincere thanks to RAP for their partnership and continued dedication to advancing education in rheumatology. We would also like to thank Johnson & Johnson for supporting this activity. Now, let's begin. Monica, to begin our discussion, could you walk us through the structure of interleukin-23 and the receptor complex?
Sure. The IL-23 cytokine family are unified by a shared P40 component, but they have different functions. In blocking P40, we block both IL-12 and 23. There's a separate P19 component that allows us to block IL-23 without blocking IL-12. The IL-12 component activates Th1 cells, while the IL-23 component predominantly activates Th17 cells. These agents then bind to their respective receptors, which activate intracellular signaling via the JAK-STAT pathway to mediate an inflammatory response. IL-12 and the Th1 pathway activate interferon gamma, which is a key inflammatory mediator. However, by blocking IL-12, we prevent the production of interferon gamma, which is also important in protecting us from intracellular organisms and viruses. IL-23 and the Th17 pathway activate IL-17A, IL-17F, IL-22, and TNF-alpha. By blocking IL-23, we mitigate conditions in which the IL-23 pathway and these downstream mediators are overexpressed, specifically the spectrum of psoriatic disease, psoriasis, psoriatic arthritis, as well as inflammatory bowel disease with Crohn's disease and ulcerative colitis.
In terms then of the immunology, how does interleukin-23 influence directly Th17 cells and cytokines?
IL-23 is an important mediator in psoriatic arthritis. From a pathophysiologic perspective, in a genetically susceptible individual, classically thought to be HLA-B27, but newer data tells us there are other genes that are potentially involved, for example, HLA-B8, HLA-B38, and B39. There's some sort of environmental trigger, whether that's an infection, whether that's mechanical stress at an enthesis, or at an enthesial point, whether that's microbial dysbiosis in the gut, there's some sort of trigger that activates dendritic cells to release IL-23. IL-23 is what's known as a proximal cytokine. It then activates Th17 cells to release what are known as effector cytokines, IL-17A, IL-17F, IL-22, and TNF-alpha. And these mediators are responsible for the clinical phenotype we see in psoriatic disease. However, we can also see IL-17 production from the innate immune system, which occurs independently of IL-23. This is a hypothesis as to why IL-17 medications, but not IL-23 medications, are efficacious in diseases such as axial spondyloarthritis or ankylosing spondylitis, because it is felt these diseases are more driven by the innate immune system, whereas the adaptive immune system is active in psoriatic arthritis. Taking a step back, if we look at the cytokines, cells, and organ systems involved in psoriatic disease, there is a redundance of the immune response. Even though we think we understand, this is likely an oversimplification.
As we focus on potential therapeutic targets, which Jessica will cover, from an immunological perspective alone, the Fc portion of a therapeutic antibody may or may not affect the function of that antibody as a therapeutic target. Do you think that this is relevant?
Very much so. Much like TNF inhibitors, where we have 5 different FDA-approved therapies, we have 5 different FDA-approved IL-23 medications across all disease states. While these molecules all inhibit IL-23, analogous to TNF inhibitors, they have different structures. And one aspect of these differing structures is the Fc binding portion, which has implications at the cellular level. The structure of the Fc portion of the IL-23 antibody may allow it, or not, to bind to the CD64 binding site on monocytes. Some of the IL-23 medications, for example, guselkumab, have a wild-type Fc binding portion, and that allows them to bind the CD64 site on monocytes. Other IL-23 medications, such as risankizumab, have a mutated Fc binding portion, which does not allow them to bind CD64 on monocytes. Why is this relevant? This is relevant because monocytes are a major producer of IL-23. It's been shown that over 75% of IL-23 production in skin biopsies of psoriasis patients is due to monocytes. In this way, there's a potential dual binding capacity that some of the IL-23 medications possess, where they bind to the P19 or P40 subunit on IL-23 versus via the FAB region, as all of the IL-23 molecules do, but some are also able to bind at the CD64 site on monocytes via their wild-type Fc receptor. From a structural perspective, there are also differences in terms of binding affinity to IL-23 among the class. It's important for us to understand the different structural differences among these molecules.
The development of P19 selective monoclonal antibodies marked a shift in therapeutic precision. Can you explain the rationale behind targeting P19 rather than P40 and how this differs across agents?
As discussed earlier, the IL-23 pathway is critical in psoriatic disease. Targeting IL-23 via the P19 subunit without inhibiting IL-12 via the shared P40 subunit and subsequent downstream Th1 pathway provides a more selective therapeutic approach. This can help preserve Th1-mediated host defenses against intracellular organisms and viruses. Maintaining IL-12 activity may help reduce autoimmune severity, as IL-12 may have an immunoregulatory function in late-stage inflammation. It's also thought to preserve anti-tumor surveillance, as well as promote bone health, as IL-23 promotes inflammation-induced bone loss through osteoclast activation, while IL-12 may inhibit it.
Thank you for that complete explanation on the immunology of what we now understand for the IL-23 molecule. Now, are there questions that you're interested in asking from that presentation, Jessica?
Yeah, sure. So Monica, given the complexity of the immune response, do you anticipate there being a role of IL-23 in spondyloarthritis?
Great question. Thank you. So classically, the IL-23 medications have not been successful in classic axial spondyloarthritis or ankylosing spondylitis. However, more recently, we've been identifying and defining axial disease in psoriatic arthritis, in particular, whether that behaves similarly to the classic axial spondyloarthritis or whether it is its own unique entity. And there are differences among these two entities from a genetic perspective, from a demographic perspective, from a radiographic perspective. So I would argue I do think these are distinct, and there are current efforts underway to formally assess the role of IL-23 in axial psoriatic arthritis through a trial known as the STAR trial, which looks at guselkumab in psoriatic arthritis patients with axial disease.
It's interesting. 3 of the IL-23 agents have been tested for the treatment of axial spondyloarthritis, not psoriatic arthritis alone, and all 3 failed. Guselkumab was the only one that was not tested in axial spondyloarthritis. So I think that this trial will be interesting in 2 regards. Number 1, does interleukin-23 therapy work for axial PSA? And number 2, and probably more importantly, is axial psoriatic arthritis a separate entity? I have one other question that I think I would be interested in hearing your answer. Given the complexity of the immune response that you just described, is there a rationale for targeting 2 cytokines or targeting a cell type and a cytokine for the management of psoriatic arthritis?
Certainly. There are patients resistant to multiple therapies. As a field, we're currently wrestling with how to define these patients, the so-called difficult-to-treat psoriatic arthritis. First, it's critical to differentiate active inflammatory disease from fibromyalgia and from osteoarthritis. And in the patients with active inflammatory disease recalcitrant to multiple therapies, it may make sense to combine therapies targeting multiple different pathways. There are efforts underway to formally study this. Affinity is a clinical trial assessing the efficacy and safety of dual therapy in psoriatic arthritis using 2 therapies with distinct mechanisms of action. It looks at combining golimumab, an anti-TNF-alpha medication, and guselkumab, an IL-23 inhibitor, in patients with active psoriatic arthritis. So I think this is something we will have more information on in the near future.
Thank you very much to both of you. This concludes part 1 of our 3-part series on the IL-23 paradigm in psoriatic arthritis. We hope this foundation discussion on the immunology of interleukin-23 has helped illuminate why this cytokine has become such a pivotal focus in PSA management. Be sure to join us for episode 2, where we'll shift our attention to a detailed review of the currently available interleukin-23 inhibitors, including their mechanism of action, clinical trial data, and differentiating characteristics. If you found this episode valuable, we encourage you to follow or subscribe to Across the Rheum, the official podcast of Rheum Connect or the Arthros website. Thank you, Jessica Farrell and Monica Schwartzman, for being here with us. We look forward to continuing the conversation. See you in episode 2.